Next speaker is Meng. Her report is about the Genome project. So we want initiate global Syngnathiformes genomes as DSD projects. So, but sometimes it's really difficult to get some big data, so after a while, maybe Meng can give us some outline about what we do in the next. So let's welcome.
Good morning, everyone. I am Meng from Professor Lin's lab, South China Sea Institute of Oceanology, Chinese Academy of Sciences. I'm really happy to have this opportunity to introduce the Global Syngnathiformes Genome Project. It is an international cooperation project for genome sequencing and data sharing. All of you are warmly welcome to join this project, and any suggestions are appreciated. The order Syngnathiformes is a large and extraordinarily diverse clade of ray fin fishes with 11 families, 91 genera, and about 670 species of seahorses, pipefish, and their relatives. Lineages in this order usually have specialized morphologies and were often referred to as the bush at the top, and also thought to be the Darwin's and least forms most beautiful. For example, they have many unique traits, such as the tubulous note, which is the source of their Latin name Syngnathiformes or Syngnathidae, means the jaws get together. And their extended body is often covered with an armor of bony plates instead of the scales. The family Syngnathidae is thought to be the only family in the animal kingdom that display male pregnancy, and their broad pouch structures is accomplished varies between species. Oh, sorry, the PPT is delayed. This group is widely distributed in the tropical and warm temperate regions all over the world. The origin and the mechanism shaping the radiation of Syngnathiformes is now one of the hotspots of evolutionary biology research. However, many species in this order may be at heightened extinction risk because they produce relatively few offspring and exhibit high site fertility. In addition, more than 1/3 of the species are considered data deficient or even not evaluated, so we cannot know their extinction risk. More research on the biology and the conservation need to be conducted for saving these special and beautiful fishes. In the past decade, we have witnessed the rapid development of the genome sequencing technology. And the first genome of Syngnathiformes species was published in 2016 as the cover paper in Nature. By sequencing and analyzing the genome of a male tiger-tailed seahorse, the author found that the seahorse evolved at a faster rate than its ancestor, leading to key genetic changes. Seahorses lack the gene TBX4 for the pelvic fins, and also have an abundance of piston genes, which is associated with the male pregnancy. In the same year, the genome of the dove pipefish was published subsequently. The authors found that the gene loss and the changes in the hawks and the DLX clusters were candidate mechanisms for the evolution of synaptic trails, including the elongated axis and the loss of the ribs. The authors also noticed that the piston genes and male pregnancy. The first two genomes of seahorse and pipefish helped us to explain why the synathate looks the way it does. Last year, the genome of a trunk brother, Manado pipefish, was published. Comparative genomics reveals the signatures for genetic changes are convergent between synathate and placenta mammals, and the loss of most PQ-rich SCPP genes in the synathate might lead to the loss of teeth in this lineage. In the same year, an outstanding paper published in the PNAS sequenced 12 genomes of Syngnathiformes species in 6 families. This paper uncovers a modification of the adaptive immune system in seahorse and pipefish that coincide with the evolution of male pregnancy, indicating a trade-off between immunological tolerance and the embryo rejection to accompanying the emergence of male pregnancy. This year, On the basis of almost 360 seahorses genomes from 21 species, researchers studied how these special fish were able to spread so successfully worldwide. Based on an evolutionary tree, it was possible to reconstruct the disposal routes of the seahorses worldwide and to explain where and when new species emerged. This study also reported the first chromosome-level assembly genome of the Syngnathiformes genomes in the light seahorse. Besides the above mentioned, there are some other Syngnathiformes genomes that already released in the NCBI or already published in the preprint. Due to the success of comparative genomic studies in Syngnathiformes, we decide to expand our understanding and exploration of this unique lineage of fish, and also extend these findings into multiple disciplines. To do so, international cooperation must be a priority. So here, we announce the GSG project. It is an international collaborative project aiming to sequence about 300 to 500 Syngnathiformes genomes within 5 years. And we officially welcome all collaborators all over the world to join this effort. The sampling should be at least one species for each of the 11 families as the first step, and then cover as many genera or species as possible, especially species in the key evolutionary nodes. For the key species, they will be sequenced using the third generation long-read sequencing along with the high-C sequencing. So we can get the chromosome level assemblies. And by comparative analysis, we can address various questions such as the Syngnathiformes genome evolution, the diversity formation, male pregnancy, and their unique morphologies. In addition, we want to set up a database for data sharing in which all the genome data will be uploaded and keep updating. And the purpose of this project is to address questions for Syngnathiformes, including evolution, biology, resources, conservation, exploration, and utilization. The GSG project will start in the beginning next year. So from now to the end of this year, we can discuss and set the detailed rules of this project. Again, all of the collaborators all over the world are warmly welcome to join this project. And we envision that this project will have a significant scientific and public impact that will also change our understanding of the Syngnathiformes species. Thank you very much.